All right, so we're going to continue with the lecture on DNA replication. And where my previous video cut off was for elongation of DNA. And basically, after the initiation aspect of replication, there's a restriction imposed by the DNA polymerase itself. And this results in the leading strand and the lagging strand being synthesized in two different ways. Basically, what I mean is the two different strands of DNA will get synthesized differently because they are anti-parallel. One goes in the 5' to 3' direction, the other 3 to 5'. So the leading strand is the one that allows for continuous synthesis towards the replication fork. It's being read from 3' to 5', allowing the polymerase to act in its preferred direction. It writes the new code from a 5' to 3' direction, so it can just follow along since it's anti-parallel. As the replication machinery unzips further at the replication fork, the polymerase can just follow. Now, the lagging strand requires discontinuous synthesis, meaning away from the replication fork. That's because the 5' end is over here on the opposite side of the 5' is over here on the opposite side of the 3' end. The 3' end is the one by the fork. So we have to attach our polymerase here by the fork, and it moves in the opposite direction away from the unzipping replication fork, excuse me. So as a result, when the polymerase gets to the end of this fragment, the helicase will have gone further past where the replication fork was at the beginning. So the polymerase will have to disassociate, come back to where the DNA has now been unzipped, and then copy in the direction until it hits where it started before. So this is called the lagging strand because the polymerase is going to be lagging behind the opening of the replication fork. And as a result, all of these little pieces that it makes are called Okazaki fragments for the husband and wife couple who found them. And then over time, these Okazaki fragments are going to have to get stitched together to keep our DNA from having single-stranded breaks in it. So the cell, to do this, is going to coordinate the activity of two different DNA polymerase III enzymes, the one going towards the replication fork and the one going away, the one on the leading strand, the one on the lagging strand. And with primase and helicase, they're going to make sure that both of these strands are being synthesized and being elongated simultaneously, even though the lagging strand is quote unquote lagging behind. In the end, its synthesis is nearly just as fast as the leading strand. Excuse me. This is possible because as this is all happening, the polymerase that's going on the leading strand will just stay in its associated place in the fork, and so will the one on the lagging strand. They will both stay right across from each other in the replisome. This causes the newly unzipped DNA to kind of push out and form this single-stranded part of DNA that's looped out of the complex. And this is going to have to be stabilized, which we'll talk about in a moment, and then the polymerase will let go of the strand, grab where the single strand is started, and then fill in the gap. Now, as the synthesis is proceeding, DNA gyrase is going to be operating ahead of the fork to relieve that torsional strain. They've just omitted it from most of the pictures because it was getting complicated, but you can see it right here doing its job. So let's talk about this a little bit more. The single-stranded DNA is a target for degradation. So if we don't want this single-stranded aspect to be broken down while the polymerase is making its current Okazaki fragment, we are going to have to stabilize it. And we do that with single-stranded binding proteins. They come in and they're going to stabilize the DNA and prevent it from being degraded. Excuse me. Um, until the polymerase is ready. Then it will detach, attach back up at the start of where the single strand is beginning from the replication fork, and then it will fill in the gap and just knock off the single stranded binding proteins as it goes. And it will end its replication when it hits a previous Okazaki fragment. Now, when we're elongating, there are primers that have to be made first at the first starting point of the leading strand and at every single Okazaki fragment on the lagging strand. So these primers have to be removed by either RNase H or the exonuclease activity of DNA polymerase 1. And that's because we want DNA, not RNA. So we need to remove any little pieces of RNA from our DNA and replace them so we can have a continuous DNA molecule. So the exonuclease activity of DNA Pol 1 or different RNases will come and take away that whole RNA primer. And now DNA polymerase 1 has a template that it can synthesize DNA from. And it will fill in that patch of DNA, but it won't end up linking that phosphodiester bond in that last moment. There will still be a single stranded nick in the DNA itself. So we have to have one more enzyme come in, DNA ligase. It repairs that nick using energy from NAD in bacteria or ATP in eukaryotes to basically add a phosphate group in there and form that phosphodiester bond. So let's get into termination now. This is bidirectional replication, so we have two different replication forks going in opposite directions of each other, and they're going to meet at the opposite side of the chromosome. There, they're going to find a bunch of terminator sequences, about 10 of them. They're shortened with TER, and this is on E. coli in particular that we're talking about here. This is the same way it's done in pretty much all bacteria, but we use E. coli as the model. And their job is to stop the replication fork, and it keeps the polymerases from continuing on and just keeping on copying this over and over and over again or running into each other and crashing. So a protein called TUS, the terminus utilization substance, is going to bind to these terminator sequences. And when that happens, the DNA polymerase won't be able to continue past them. It'll bump into this protein that's associated to the DNA and not be able to continue and often disassociate. Now, there's many different terminator sequences in case one of them doesn't work. It can be caught by the next one. But how we maintain these terminator sequences in future replications is still kind of unclear. The whole ending part of replication is still being looked at by researchers today. So there is a video of this. I highly, highly recommend you guys watch this one because replication is very difficult to show you on flat two-dimensional images. When you can see it moving and all the pieces coming together, it clicks a lot better. Now, in circular chromosomes, after replication, we run into a really interesting problem. The two sister chromosomes, the two identical chromosomes that were just copied, are linked, kind of like a chain link fence. And we call this a catenane, the pair of the linked ringed chromosomes. And this comes from the Latin word for chain, like in Italian, you say cadena. So there you can see that this structure has to be resolved in order for the two sister chromosomes to separate and the cell to correctly divide and have a chromosome in each new daughter cell. So this has to be resolved, and it gets resolved by topoisomerase 4. It comes in and introduces a double-stranded break, passes one of the chromosomes through that double-stranded break, and then we resolve the double-stranded break, and now they're separated. Now, there's also instances that can occur called homologous recombination. This happens in eukaryotes too. If you remember studying meiosis, when the sister chromatids on two different chromosomes get close to each other, they can what we call cross over, and you can have one piece of one chromosome detach and jump onto the other chromosome and have that piece of the chromosome, number 2, break off and go back to number 1. So you end up with a piece of the other chromosomes swapping places. And this happens anytime you have what's called homologous DNA sequences, DNA sequences with the same overall sequence. Maybe there's a couple differences in there, but pretty much those sequences look identical, excuse me. So when you do this on a circular or two circular chromosomes and you have a homologous recombination event, if you start at one origin and you now follow the chromosome all the way around through that recombination event, you can see that we've now just created a super chromosome. It's one entirely linked molecule now. And we don't want that. We need them to be separated because the goal of replication is to put a new chromosome into each new daughter cell. So we have instances or enzymes that can fix it if this happens. It's not super common, but it does happen. And there's termination factors, in particular FITZ-K, ZIR-C, and ZIR-D. And what they do is they come in and they create a second homologous recombination site somewhere else on the chromosome, and that effectively cuts them and creates two more chromosomes again. But you will notice that we had a piece of each chromosome now switch places. So a piece of chromosome 1 is now on chromosome 2, and the same exact part for chromosome 2 was put onto chromosome 1. And then we discussed this in class a little bit, but be able to answer a question like this for the exam. On top of the primary chromosome, bacteria can have plasmids and secondary chromosomes. These are extra chromosomal DNA elements. They replicate autonomously, so they replicate themselves, and they often contain extra genes that might be useful to the cell. So let's dive into these. So plasmids vary in their size. They can be smaller, they can be quite larger. They vary in their copy number, so how many copies of the plasmid exist in the cell at any time, and their cargo or their content. So what genes are found on the plasmids varies from plasmid to plasmid. And these are found in all domains of life, not just bacteria. They are typically much smaller than the chromosome. They're usually circular, they're usually negatively supercoiled, just like the chromosome itself. And they'll often, or they have to contain non essential genes. If they contain essential genes, then we consider them a chromosome, not a plasmid. So, while these genes are non essential to life, meaning the bacteria can survive without them, the genes that these plasmids bring often bring in a critical role. They help the cell in one crucial way, such as antibiotic resistance, and this makes the cell not want to give up the plasmid. It'll tolerate the existence of the plasmid in the cell. Now, plasmids are transmitted between cells in three different ways: conjugation, transformation, and transduction. And we'll discuss all of these in much more depth later. But basically, conjugation takes a plasmid from a donor cell, copies it, and gives the copy to the recipient cell. In transformation, bacteria can pick up DNA from the environment that does not belong to them. And in transduction, viruses are responsible for moving DNA from one cell to another, because they accidentally package up the DNA of another cell rather than the viral DNA into their capsid. So, how do plasmids replicate? They will regulate their own replication. So, there's two different ways that they can do this: bidirectional replication, which is where they start at a single origin and it goes in two opposite directions simultaneously, just like what we learned with DNA replication in general. And then there is rolling circle replication. This is going to start at one single origin. We're going to create a single stranded nick in the DNA, and we're going to copy it as we move around the chromosome. Now, plasmids will borrow the machinery of the host to copy themselves, but they'll have their own origin sequence, their own initiator proteins, and they will control the frequency of their replication. Now, Rep A is a protein that binds to single-stranded DNA. So it's going to bind to one single strand of the DNA in the plasmid and create a single-stranded break. We call that a nick. And the other strand is going to be left completely intact. So we break one strand but leave the other alone. Now, the 5' end of this broken strand is going to be bound by rep A, and that's going to leave the 3' end open for a DNA polymerase 3 to bind and begin replication in the 5' to 3' direction. So it's going to use the intact strand as a template. Now, one of the double-stranded, um, it's going to end with one molecule being double-stranded and the other being single-stranded. That's going to get moved into the other cell, the recipient cell, and in the recipient cell, it's going to be copied like normally. So I'm not going to test you on the exact mechanisms of rolling circle replication. You have to know what it is, but I'm not going to make you explain all the parts to me, but there is another video about that if you'd like to learn more. So now plasmids can also regulate when they replicate, not just, or how many copies of themselves that they make is what I mean by that. There can be single copy number or multi-copy number plasmids. In single copy number plasmids, they're only going to replicate when the chromosome itself replicates. This means that there's never more than one of these plasmids in the cytoplasm at a time. Then there's multi-copy number plasmids, which they will copy themselves as many times as they can. That way they flood the cytoplasm with as many of these plasmids that randomly will get taken up by one of the two cells basically during binary fission. The idea is if I make enough copies, one of them is bound to get into each new cell. Um, so we are not going to go over the handcuffing in this class. We're just going to focus on the plasmid copy number idea. Now there are tricks to ensure that the plasmid is inherited. They will have many different strategies to make sure that they get into the new host cell. Some genes will carry really important, or some plasmids will carry really important genes on them, like antibiotic resistance, and this makes the cell not want to lose that particular plasmid because it's giving the cell a selective advantage. Now, there's also structures that can be formed within the cell that are kind of like cytoplasm or cytoskeletal structures, and they can form and push the two copies of a plasmid to opposite poles of the cell to ensure that when the cell septates, one is in each new daughter cell. Now, secondary chromosomes evolved from plasmids. These are chromosomes where they are smaller than the primary one, but they carry at least one essential gene. If that gene happened to go away because the cell decided we're not going to keep this secondary chromosome, the cell would die. It wouldn't be able to maintain itself. So there's evidence that these secondary chromosomes were once plasmids and they somehow captured the gene from the chromosome itself, one or more genes, and brought them onto the plasmid and created a second chromosome. And that is it for this little catch up from when my microphone went out. I apologize for that. If you guys have any questions, please do not hesitate to reach out to me, and I would love to help you. Hope you're having a great day. Talk to you guys later.